Hello, everyone, and welcome to today's webinar, Latest Technologies in Antibody and Protein Labeling. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Douglas Anderson, Scientist 3, Chemistry R&D, Thermo Fisher Scientific. Doug, you may now begin your presentation.
All right, thanks for that great introduction. We'll go ahead and get started. My name is Douglas Anderson. I'm a research and development scientist here at Thermo Fisher Scientific, and I'm going to be going over some latest technologies in antibody and protein labeling. Before we get started, I wanted to overview what we're going to cover in today's webinar. We're going to start with some basics just to make sure everybody's on the same page, some general information about conjugation basics, how the workflows come out, types of applications you might be using conjugation for, and then moving on to some more nitty-gritty with one of the questions I get all the time as a conjugation chemist, which is, how do you select conjugation approach and how do you pick a fluorophore? What is the right conjugation for you, whether you're going to create your own conjugate in the lab, using a kit or freehand, or if you're going to purchase a conjugate for your experiment. And then lastly, I'm going to be covering some of the state-of-the-art improvements in conjugation chemistry, both some products here at Thermo Fisher Scientific, some workflow improvements, and some products for purchase that have really pushed the boundaries of the conjugation chemistry in general and push the bounds of what you can do at the bench and what you can do in your experiments to really get you premier reagents to give you the best success in your next round of experiments. So diving right into it, what is conjugation? So conjugation is the technical term we like to use that really just refers to any kind of biomolecule labeling. So conjugation is a series of chemistry techniques that in general we use it to refer to any kind of labeling of a biomolecule. This is most often an antibody or a protein of interest, but it can be really any kind of biomolecule, so nucleic acids, any kind of biopolymers, glycans, dextrans, really anything in life sciences that you want to attach some kind of label to this biomolecule. And this label can be really almost anything that suits your application. It's most often a fluorescent label or perhaps a colorimetric label, but it can also be small molecules, haptens, biotin, other proteins, or crosslinkers. Really the sky's the limit as far as what you want, but everything in that we call conjugation shares in common some kind of a labeling of a biomolecule that you're going to be using in some kind of downstream application for an experiment. On the right here, we have an example of what an actual conjugate looks like. So this, for me, I'm very visual, and I really enjoy thinking about what an actual bioconjugate would look like. So this here in this example of a crystal structure model of an antibody that's been labeled with Alexaflor 647 to give you an idea of what this looks like here. The green residues are highlighting lysines, which are the particular target for this fluorophore. So moving on to what we know what a conjugate is, how do you make them? One of the reasons I like to start here is, if you're unfamiliar with bioconjugates, people often have an idea that they're quite challenging, and in reality, they're actually, for the most part, fairly easy to make. But generally, producing a conjugate, whether you're making one from scratch or buying a kit, is pretty straightforward. Most conjugates are made in about 3 or 4 steps. You have a general prepping some reagents for use in the reaction that you'll be performing. Most of these reactions are simple one-step reactions that take place between 1 to 3 hours at room temperature, so nothing too fancy, followed up by generally a single-step purification to remove any excess labeling reagent. And then finally, a quantification of that biomolecule that you've created before it's ready for use. So in general, depending on the label and the workflows that we'll be talking about today, Most conventional conjugates you'll find on the market can be made within a few hours, and depending on the workflows, they can sometimes take a little longer if you're making a more complicated workflow or more complicated reagent. But most of the conjugates you'll see on the market and reagents for are relatively straightforward to make yourself if you're wanting to make one yourself for your next experiment. So before I move on, I'm going to be talking about a couple general conjugation chemistries here to highlight the types of chemistries and the approaches that you can take when making a bioconjugate. But I do want to point out that this webinar is not intended to be a super detailed overview of everything that's out there. There's quite a lot of great resources out there if you'd like to really dig into the nitty-gritty of all the various chemistries that are out there and the different types of approach. So I'd like to point out the Molecular Probes Handbook. It's actually made Thermo Fisher Scientific for free. You can get one of these copies online, and this goes into all the various details of all the different types of chemistries, all the products that are out there, how to go through a conjugate, all the interesting details between the different reactive groups available. There's also a textbook, an excellent textbook, Bioconjugate Techniques by Greg Hermanson, that is literally the book on bioconjugation that has all the detail one could ever want as far as how to attach a label on a biomolecule. Lastly, there's also another webinar from a colleague of mine that was made in 2016 that's available for free on YouTube, where she describes in full detail all the various conjugation chemistries in all their glory. But for here, I'm going to stick to kind of the four most popular conjugation techniques and just really cover the general conjugation chemistries that you might see in a conjugate or they might use in the lab. So in general, there's four classes of conjugation chemistries, and it's easiest to think about how these conjugation chemistries, what are they targeting, what are they reacting with on the biomolecule. This will really help determine what type of approach you might want to use or what type of bioconjugate you might want to purchase if you're going to be buying them. They have different pros and cons, which we'll actually go to in a few slides here, but I do want to highlight these four general categories of the types of chemistries that they're doing. And these can be generally broken down into four categories. So you have your amine targeted, so these are reactive compounds that are targeting the lysines and proteins or any free amino groups if you're looking at a dextran or a glycan. You have sulfhydryl targeted reactive groups, so these are going to target the cysteines and proteins. Then you have some cross-linking reactions that are generally used to cross-link proteins to make protein-protein conjugates. So these are designed to have controlled cross-linkings to give you well-defined conjugates between large proteins such as APC, RPE, GSP, or enzymes of interest to produce protein-protein conjugates, and often these are antibody conjugates to either an enzyme or a large protein fluorophore. Lastly, you have one of the newer categories, which is click chemistry. This is probably one of the newest ones that we have on the market, and I've actually got a couple slides dedicated to some of the recent advancements in click chemistry. So I'll step through these four categories in brief and highlight kind of what are the reactions that we're looking like, what are the reactive groups before we move on to the pros and cons. So the first group, the amine targeted, this is by far the most common and popular strategy for labeling a bioconjugate. So these reactive groups target, as I mentioned, the lysines of proteins, and these are usually simple one-step reactions, and the most common of which is what's called an NHS ester. So these labels, usually fluorophores, will have this simple NHS group that will target the lysines on a given protein of interest, and you have a simple one-step reaction at mild alkaline conditions. And after about an hour at these conditions, you'll be left with a stable covalent amide bond between your label of interest and your protein. And then you'll have the leading group, the NHS group, to be purified off in downstream purification. These are by far the most common conjugates produced. They're very stable. They're easy to produce. The reaction is very well characterized. And they're easy to do as an end user if you just want a straightforward conjugate. There are other reactive groups that you might see on the market. Generally speaking, they all more or less do the same thing, some of which are a little less susceptible to water hydrolysis, but in general, all amine-targeted chemistries like NHS ester, SDP ester, PSPs that you'll see on the market all more or less target the amines in the same way and have similar reaction conditions. Second most popular conjugation chemistry group would be sulfhydryl targeted. So as I mentioned, these are reactive groups that are targeting the cysteines on proteins. So these reagents here react with the reduced sulfhydryl groups of cysteines in proteins. These, similar to the amine targeted, these are usually a simple Malayamaid reactive group. So it's seen here, very similar to the NHS group, with just that extra double bond. These react with reduced sulfhydryls at a neutral pH in about an hour, sometimes a little bit more, hour and a half, 2 hours, to produce a stable thioether bond. So these sulfhydryl chemistries also very easy to do at the bench. Quick reaction times, well characterized. These chemistries have been around for a long time. So also have a very nice, easy workflow of a single one-shot reaction followed by purification. So another good classic choice to put a label on a biomolecule. The third group here is a little more general. I've called it the protein-protein cross-linking group. So if you have the need to put a protein onto another protein, so most often an antibody conjugate with either a enzyme of sort, often it's another fluorescent protein conjugate. So in this case, what you're trying to produce controlled cross-linking event between two large proteins. So there's a number of reagents out there that are designed to produce these well-controlled cross-linking. I emphasize well-controlled because if they're done, if the stoichiometries are off or if they're poorly controlled, you can get too much cross-linking and you'll end up with just a large protein goop, which won't do anybody any good. So you'll have this family is more about the cross-linkers themselves. One of the most common cross-linkers that you see is actually a combination of the first two chemistries. So one example here is a sulfo Sncc cross-linker, and this is basically an NHS ester with a short linker itself, a linker of linkers really, where you're just linking a amine targeting NHS group with a sulfhydryl targeted Malayamaid. And so you can basically pop one side of the linker onto your first protein under one of the conditions for one of the reactive groups, and then you follow it up with your second protein and you use the second reactive group's reactive conditions. And this two-step method lets you have a well-defined cross-linking event where you can control the amount of cross-linking between the two to give you a well-characterized protein conjugate. And the last group I want to talk about here is the click chemistry. So this is a newer group of reactions, reactivities for bioconjugations. And this is actually a two-step process that involves a reaction between a azide modified biomolecule and then a cyclooctane modified biomolecule. And the benefit of these using click chemistry between this azide and cyclooctane is that this is a highly efficient bioorthogonal catalyst-free reaction. And one of the major benefits to this type of conjugation is it can actually occur in vivo. So you can use it to create stable conjugates in test tube, of course, but there's a whole product lines involving using click chemistry in vivo as well. These reactions are, again, highly efficient, so they usually go to full completion, and sometimes purification is not even required. And in this reaction scheme here, no catalyst is needed either, so it's very gentle on the conjugate. And I'm going to be providing a little more detail in a few slides about our Cyclic technology, which takes this chemistry to the next level to really give ideal conjugates. All right, so now that we have a general overview of what is conjugations, how do you perform conjugations, one of the major questions I get as a conjugation biochemist all the time is, how do I pick a conjugate? What is the right conjugate for me? What's the right conjugate for my application? I always, when I'm answering these questions, I really ask, what is your application? What is as an end user, what do you need? And the different chemistries and fluorophores available will have different suitabilities depending on your application. So some of the most common applications for a bioconjugate, often an antibody, fluorescent conjugate. I have 4 applications listed here. There are others, of course, and your needs may vary. But in general, most conjugates fall into either immunocytochemistry, where you're trying to take a picture on a microscope, flow cytometry with primary fluorescent labeled antibody conjugates, antibody drug conjugates for research use in ultimately coming up with IVDs or perhaps an in vitro assay. So walking through these 4 conjugation chemistries, what are they best suited for? So with amine labeling, as I mentioned when going over the general conjugation chemistries, this is a very tried and true labeling method. It's extremely straightforward and also inexpensive, as this has been on the market for 30, 40 years and is really the gold standard when it comes to antibody conjugates. These conjugates, while they're straightforward and relatively simple, they more often than not are able to meet the needs of kind of your workhorse applications. So that'd be immunocytochemistry, perhaps if you're making a secondary conjugate, or if you want to go with a one-step primary conjugate. Even though the technology for this is relatively, has been around in the market for quite some time, these conjugates are often more than bright enough for most applications and are really the gold standard when it comes to most labeling experiments, whether that's a simple ICC stain, a flow cytometry, or an in vitro assay. One sometimes can be a con of this method is that because these reagents are targeting the lysines on proteins, depending on the protein size, there is often dozens of these lysines. So that means when you're making a conjugate, these reagents are randomly labeling the lysines on these proteins. So for a given antibody, most antibodies have around 50. So if you have a conjugate that has 3 or 4 fluorescent labels, for example, those could be anywhere on the antibody. And depending on your application, that often doesn't matter. But sometimes, especially if you're trying to make, let's say, an antibody drug conjugate, or you really want to have that the fluorophore ratio well controlled, this labeling technique might not be as precise for that technique. So again, these make excellent workhorse conjugates. If you're doing kind of traditional workflows that are well established, these are the conjugates for you. These are been the gold standard for life sciences research for decades now, and they still have lots of use in today's experiments. So next up, we have the Malayamaid sulfhydryl labeling. So this is kind of an interesting chemistry for applications. So as I mentioned here, because these conjugates or these reactive groups target the cysteines in proteins, most proteins have significantly less cysteines than they have lysines. So what this means is that even though the cysteine labeling with these reagents is still somewhat random in the sense of what cysteines it hits, because there's significantly less cysteines, you generally have a good idea of where these are going to go based off the sequence of your protein. So if this is an antibody, the cysteines are well mapped out. So you more or less know where your cysteines are going to be. So this makes for a good application for conditions where you really want that control over the labeling. You want to know where your fluorophores or your label of interest is going to be. So it gives you that labeling control. And because it's hitting a much smaller pool of cysteines, you have improved repeatability just simply because there's less options for them to label. So you're going to have more of a lot-to-lot repeatability between labelling. So you're not going to have nearly as much of that randomness. And these protocols are just as easy to follow as a amine-targeted reagent. So you have a nice workhorse labeling that is easy to do at the bench, but gives you a little bit more of a well-defined conjugate. One of the big cons, though, of a sulfhydryl targeted reagent is that because you're targeting these cysteines, which are usually significantly less than the lysines, most of these conjugates are going to have significantly less fluorophores per antibody or per protein. So they often have a lot lower signal to noise than a traditionally labeled conjugate. So you really want to, if you're looking at implementing this into your workflows, you really want to consider, is the trade-off of signal, the potential for lower signal, worth it for that extra control? And this really becomes really useful in vitro assays, where if you're using a protein where you can genetically manipulate the sequence, perhaps you can add a cysteine at the end, at the end terminus or the end of the protein. If you can control the location of that cysteine, you have great control over exactly where you're putting that fluorophore, so it might not interfere with your protein. So if you have an in vitro assay where you can have that level of control, this is a great method for you. And it's also great for if you're trying to make a defined antibody-drug conjugate where repeatability is quite important, and maybe you're not putting a fluorescent fluorophore, you're putting some kind of a drug where you only need one or two of those. So this is a great method if you need that control and that repeatability above all else. All right, next up we have our click labeling, which I mentioned earlier. So click labeling has really come into its own as one of the premier labeling technologies. And I'm actually going to spend a couple slides dedicated solely to talking about Cyclic, which is one of our premier labeling technologies at Thermo Fisher Scientific. But click chemistry has really become quite useful in making really exceptionally well-defined conjugates that are very well controlled and very well repeatable, with also efficient workflows. So in most click chemistry, especially our Cyclic platform, these workflows provide very well controlled labelings, as the labeling is specific to a certain type or certain position on the antibody. And so this gives you exceptional labeling control. It gives you exceptional repeatability. And I'll go into a little bit more detail about our Cyclic platform here in a few slides. But in general, these click platforms that are out there on the market give fantastic fluorophores or fantastic conjugates with well-defined number of fluorophores per antibody or protein. The reactions themselves are very efficient, so there's less to clean up in your purification step, and sometimes to the point where you don't even need to do a cleanup. However, these workflows can be a little bit more expensive, and they do add a little bit of extra hands-off time performing the extra steps to generate the intermediates that are needed, such as the azide activated antibody in this figure here. So where this really becomes very helpful is if you have an application where you need a exceptionally high performing antibody, where you really need everything. You want to preserve the integrity of your antibody, you want to preserve the integrity of your protein at all costs. And this is a great method for that because it keeps your labels away from the binding sites or perhaps the biological activity of another protein. So if you really need that exceptional activity of your biomolecule preserved, Cyclic and click chemistries are the way to go. They are more expensive, however. So if you have something where a traditional conjugate might do, maybe you want to think about using traditional chemistries, as they will be significantly cheaper and faster. However, if you have an application where you really need that high performance, Cyclic click chemistry is the way to go. All right, so I spent some time talking about how to pick a conjugation chemistry. The other question I get all the time is how do you pick a dye? How do you pick a fluorophore? There's so many out there, and there's a lot of different categories. How do you know which is the right fluorophore for your application? Even though the conjugation chemistries have quite a bit of difference, even simply the fluorophores that are selected can make or break a conjugate depending on your application needs. So I wanted to spend just a little bit of time highlighting the different fluorophores that are available and how they might influence your selection based off your application. So the first category I've highlighted here is your classic dyes. So these are your simple small molecule fluorophores that have been on the market for quite some time. So your classic dyes such as fluorescing, Texas red, Pacific blue, your large protein fluorophores, RPE, APC. So these are your markets these are your fluorophores that have defined the market. I know a lot of us have probably trained on a microscope where the filters were actually named after some of these. I know one of mine has a filter that's just called Tritzy because everything was done with Texas red back then. So these classic dyes still have a use in today's market. They're generally very cheap. They're generally have good initial brightness, but they're not the brightest things out there. They've been around for 40, 50 years. They often will have acceptable signal in the beginning stages of an experiment. But if you have an application that does have the potential for photo bleaching, or maybe you have long dwell times or it's a longer experiment, these fluorophores might not be bright enough for your needs. But if you do have something where you're doing a quick measurement, maybe an in vitro assay, plenty of times these dyes, as simple as they are still more than enough signal to get the job done. But they might not cut it in more sophisticated assays. Next up, you have your second generation of small molecule fluorophores, such as our Fluoro Dyes, are developed here in Thermo Fisher Scientific. So these second generation dyes, like the Fluorophores or the Dylights, These are improved small molecule fluorophores based off of those original classic dyes, such as fluorescein. So these dyes have excellent fluorescent intensity, can be quite a bit brighter than the dyes that they were derived from. They're often very resistant to photobleaching. So if you're doing an imaging experiment where you might have a long capture time, these can preserve that signal much longer than a classic dye. There's also quite a lot of dyes out there if you have specialty needs. One great example from our Lexaflor line is our Frodo Dyes, our Frodo series of dyes. So these are small molecule fluorophores that actually have pH sensitivity. So you can really have a kind of tailor your fluorophore to your exact application. So if you have the needs to study internalization, for an example, a Frodo dye will be off at high pH and then will become significantly more fluorescent upon becoming more acidic. So if you're trying to study the internalization of an antibody, perhaps, these dyes provide you that excellent brightness and then also give you the ability to actually interrogate different biological aspects. Next up, we have our quantum dots. So moving away from small molecule fluorophores, we'll have our larger than the antibodies themselves. Quantum dots are a small nanocrystals. So these are metallic nanocrystals that are extremely fluorescent, very, very, very high signal intensity in your fluorescence output. These are larger labels than, in many cases, the biomolecules you're actually wanting to conjugate them to. But these nanocrystals are exceptionally resistant to photobleaching, almost to the point where they don't photobleach at all. They're also very, very stable. And so these fluorophores are extremely good if you have the right application to downplay their size and take the most advantage of their very high signal output. So due to their size, they can be sometimes troublesome to interrogate very small cytoplasmic targets. But if you have a target that is extracellular, perhaps, or really low abundant extracellular target, so like viral coat proteins are one great example where quantum dots can be quite useful. So if you don't have a target that is not too hard to access by an antibody, an antibody quantum dot conjugate can be a great choice that will give you extremely high signal to noise that really will let you visualize low abundant targets if that where a small molecule fluorophore just wouldn't cut it. I've actually recently made a conjugate this way where we had a viral coat protein that only had 50 copies per virus, and the only way we could get any signal and actually detect these targets was with a quantum dot. So although these quantum dots have been on the market for 10, 15 years now, and they do have limitations, but if you're in a situation where you can make the best of them, they're still extremely useful and definitely worth considering. Lastly, we have the most recent entries to the fluorophore space. We have our polymer dyes. Polymer dyes, so these are large organic polymer-based tandem dyes. So you'll actually have like a long, rigid polymer backbone where you'll have fluorophores or tandem fluorophore pairs inside a polymer. And these are relatively new to the market. They were developed about 5-10 years ago. So you have your super brights from Thermo Fisher, you'll have your brilliant violets from BD. So these types of dyes are exceptionally bright and have very long soak shifts in a tandem pair. So these are very well suited for flow cytometry, where you're making the most out of your given filter combinations. So if you have the need for a very, very bright signal, especially in flow cytometry, and you need that signal, kind of like a quantum dot almost, where you need a signal above all else, and you want to make the most of your different excitation sources in your flow cytometry filters, these polymer dyes really fit that bill. So as one example, you can have a super bright 702, for example, that's excited by your UV laser or your blue laser or your violet laser, and then you can get emission all the way into that near IR category. So these fluorophores are really interesting to work with because they really give you a lot more options in that the visible spectrum especially for flow cytometry. They do have some downsides. Even though they are polymer-based small molecules, they can be quite large and cumbersome to get inside the cell for an intracellular target. They don't always work the best for immunocytochemistry, so if you're doing a conventional cell staining, they often have issues. So at this point in the market, they're basically mostly optimized for flow cytometry, but in that application, they excel. All right, now that we spent some time on what are conjugation, what's a biomolecule conjugate, what are the different types of fluorophores and chemistry is available, now I want to get to the fun part of the talk here, where we talk about the latest and greatest and what it really is the state of the art in making your own conjugate, or how to select a conjugate for use in your next experiment here. So one innovation I love talking about from Thermo Fisher that I use personally all the time is our recently launched Zeba protein and dye removal columns. So these are a specialty resin recently and specially developed just for conjugation workflows that take a modified size exclusion resin from our Zeba product line that has been modified specifically to capture typical excess conjugation reagents. So as I mentioned earlier in the talk, in most conjugation workflows, you'll be taking a small molecule reactive dye, and you'll be reacting that with your biomolecule, and then you almost always will have excess unreacted reagents that are left over that need to be purified away before you can use that conjugate. In most workflows, you'll be using most workflows call for a size exclusion column, which depending on the workflow can be fairly cumbersome to use. These columns are specifically designed to actually capture those reagents and retain them in a pre-packed spin column. So what that lets you do is you can process large amounts of conjugate very quickly, very easily, and you can use these pre-packed spin column formats that have a 2-minute spin, and your purification is done. So basically, we sell this resin as pre-packed spin column formats, all the way from a 0.5 mil spin column, pre-packed spin column, all the way up to 10 mil resin columns. And these are fantastic if you're doing conjugations yourself. You can go from an unlabeled antibody to a purified, ready-to-go antibody in the whole thing can take about 1.5 hours. The actual purification itself is less than 10 minutes. So if you've ever conjugated with some of the older purification resins, especially if you're doing more than 1 milligram, you may have seen most workflows will call for actually pouring your own gravity column using a size exclusion and then monitoring the bands as they come through. And all of that is in the past now with these new purification resins. These are great. I've transitioned to almost exclusively using these in the lab. They're so easy to use, and they really just drastically speed up your workflow. In addition to that workflow, they also have excellent sample recovery. And we've seen in our own internal testing upwards of 90% recovery, sometimes even as high as 95% recovery, which compared to traditional size exclusion resins, which are often in the range of more like 85 to 75% recovery. Beyond the columns themselves, I'm also excited to report that we've recently incorporated these new resins into our own labeling kits. So if you've ever, we sell a variety of antibody and protein labeling kits with the popular Fluoro dyes. These kits previously had aliquots of conventional size exclusion resins. So if you've ever, as I mentioned before, if you've ever had to pack your own gravity column, we've now updated most of our kits to include this new purification resin with these pre-packed spin columns. So if our kits are now easier to use than ever. After your 1 hour labeling reaction, you simply put your crude conjugate directly onto these spin columns. You have 2-minute spins, and now you're ready to go. So this is really a huge workflow improvement, a real quality of life improvement. I absolutely love these kits. I use them myself, and we've really done away with these bulky gravity columns. They're just for 99% of the time, they're just a thing in the past. All right. I've mentioned this a few times earlier, but wanted to give it its own slide. I mentioned click chemistry and cyclic chemistry specifically, but this is a labeling technology that builds upon the benefits of click chemistry and makes it specific to labeling IgGs with basically perfect conjugate every time. So cyclic labeling technology is all about creating a reproducible, high-performance conjugate every single time with exceptional control. So in this platform, we've developed a kit workflow. We're using 2 enzymatic modifications of IgGs and I believe also IgAs and IgEs. You can make use of the SC domain glycans. So these are branch glycans on the SC region of most antibodies. And with 2 enzyme modifications of these glycans, we cleave off the terminal galactose residue using beta-galactosidase. You then add an azide group using a genetically engineered version of GAL-T enzyme. And this now incorporates an azide group that is ready to react with your favorite click-ready label. So we also have a whole library of dibo modified. So these are the reactive alkyne groups that react with the azide modified antibody. And you can have an azide activated antibody that's ready to react with any fluorophore of interest. And why this is so great is not only does it always modify these glycans on the Fc domain, which preserves the integrity of the binding sites, this also gives you complete control over the degree of labeling. So how many fluorophores per antibody do you have? So because these glycans are fixed between IgGs and IgA and IgEs, there's always 4 of these labeling sites. So if you're using a small organic dye, such as the Alexaflores, you'll typically get a degree of labeling of 4 every single time, which gives you exceptional reproducibility between batches of antibody. Likewise, if you're using larger proteins, like APC or RPE, these larger proteins will actually sterically occlude that second spot on the branched glycan. So you'll have a DOS of 2 pretty much every time, so that you have exceptional control over your conjugate that also preserves the integrity of the binding sites. So this is really the premier conjugation chemistry to use if you want to maintain the activity of your antibody and you really want to have the perfect antibody as high performance as you can make it. And then in addition, the kit workflows are all super easy to use. They add a couple overnight reactions that are all hands off, but the actual conjugation itself is a simple overnight reaction at room temperature. So very easy workflows. In many cases, exceptionally easy purification or no purification. So just a really easy workflow to follow that gives you exceptionally well-labeled conjugate, exceptionally characterized, and just really well-controlled conjugate every single time. And saving the best for last, I wanted to highlight our latest addition to Thermo Fisher Scientific, the NovaFluor platform from Phytonics, a recent acquisition and now a part of Thermo Fisher Scientific. So we're happy to discuss the latest addition to our fluorophore family, the NovaFluors. So NovaFluors are a brand new technology platform designed specifically for mostly flow cytometry that uses a DNA-based scaffold to provide tandem fluorophore pairs that are precisely tuned for various filter sets in flow cytometry and the newly emerging spectral flow cytometry. So how these work is based off of what we call phyton technology. So these are precisely controlled single-stranded DNAs that are conjugated with precise fluorophores. These then self-assemble using Phytonics' proprietary technology. They self-assemble into these double-stranded cruciform structures that we call the phytons. And using this self-assembly into the phyton, we're able to precisely control the fretting between the tandem pairs to give pretty much exactly the excitation and emission spectra that one could ever imagine, or one could ever want or need. So we actually have, next slide here. We actually have 19 different NovaFluor color combinations that have been tuned to really saturate the entire visible spectrum to provide you a conjugate that can meet any filter combination you might have in flow cytometry. Or if you're dipping your toes into spectral flow cytometry, the unique spectral fingerprint of each of these NovaFluors really allows you to have a NovaFluor panel in spectral flow cytometry and really just have so many colors all at once and have so many conditions that you can really get the ideal spectral flow cytometry experiment. And with that, we'll wrap up and I will take any questions.
Thank you, Doug, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, what makes NovaFluor dyes so suited for spectral flow cytometry? What differentiates these from conventional fluorophores?
Hey, thanks for that question. Good points there. So Novafluores have two unique properties that make them so well suited for spectral flow cytometry. So first, one of the easiest aspects is that because the excitation emission is so tunable, you can really fully populate the entire UV vis spectrum. So like because the Stokes shift is so tunable, and then the tandem, it's really kind of the perfect tandem. So you're able to really saturate the entire spectrum and have a label pretty much exactly where you'd want it based off of your filter combinations. So in that regard, it really lets you make the most out of your instrument. But the main thing on top of that is that because these are all tandem dye combinations that are precisely tuned, they all have unique fingerprints. So every Novafluor combo has a slightly different spectral fingerprint if you actually look at excitation and emission spectra. And that's one of the key differences with spectral flow cytometers, that the algorithms are really looking for that unique fingerprints in order to deconvolute the signal. So if you take a look at all the various Novafluors we offer on our Spectra Viewer tool, you can see that they're all pretty much unique, not really owed to that tandem dye pairs that's as part of the technology. So it really gives you, each color combination, even though they're all Novafluors, they have unique fingerprints in their excitation emission spectra, which allows those spectral flow cytometry algorithms to deconvolute where that signal is coming from. Great question.
Great, thank you. Our next question is, where can I get Cyclic conjugates? I haven't seen conjugates for sale with this technology.
That's also a good question. So Cyclic is one of our newer platforms. Currently, we offer conjugation kits for users to do it themselves. So we offer kits, Cyclic conjugation kits, in various size ranges, ranging from microgram quantities up to 5 milligram kits. So these are kits designed to take your own antibody, prep them within the Cyclic azide modification, and then have them ready to conjugate with a given cyclooctane label of your choice. So we offer these conjugation kits for sale, but we don't have any off-the-shelf conjugates for sale with this method, and part of that is just because the technology is so new. But we do offer custom conjugation services with our Cyclic technology. So if you're ever interested if you don't want to do the kits yourself, or you want to outsource that labor, we do have a custom conjugation service where you can custom order a Cyclic conjugate for your application.
Perfect, thank you. Next question, how long do click chemistry reactions take between DBCO and an azide, as you have shown in your slide? I have seen in the literature that it takes as little as 15 minutes, but I found overnight reactions are much more complete.
Yeah, so that's a good point. So the reaction kinetics between click handles are all slightly different, and there's actually some literature out there where the kinetics are actually defined between the different click handles. So one of the most common ones you'll see out there, the generic click handle, is the DBCO. So that's kind of a generic one. For our Thermo Fisher conjugates, we usually use dibo or S-dibo conjugates, and the reaction kinetics are actually slightly different between those. DBCO is one of the faster reacting kinetics. And it does actually, I think, as it's mentioned in the question, you actually can see the majority of the reaction done in 15 minutes. In my own hands, it's often gone to completion after 2 hours. But due to the kinetics out there is between handles is a little hard to find. So most workflows will just kind of give you a blanket statement of do an overnight reaction to make sure it's gone to completion. And that's a good general practice to follow if you're not sure about the kinetics of a given click handle. It's just safer to let it go overnight. But in the case of if you do know the reaction kinetics, like for example, DBCO is one of the faster reacting ones, it's often worth testing that yourself. In my hands, DBCO is usually done within an hour or less, but it won't be completely gone to completion in 15 minutes. Some of the other conjugates out there, like dibo, they're a little slower. So they might be done in 4 to 6 hours, and so most people will just let those go overnight because it's easier. But they do vary a little bit, but the most common workflow you'll see is overnight. But if you do want to look that up, the reaction kinetics do vary, and you can actually cut a lot of time out there if you know what your given kinetics are for the click handle.
All right, thank you. Next question. Does the click labeling method work for IgM antibodies?
So not really. So IgMs, because of that, like kind of pentamer structure, the glycans aren't accessible. So it really only works on the glycans that are present on IgGs, IgEs, and IgAs. So it's unfortunately not currently compatible with IgMs.
Okay, thank you. Next question. What is the smallest molecule you can use in the Zeba columns?
Yeah, so the columns that I highlighted today are built off of our Zeba size exclusion resin, which is actually based off of an exclusion limit of 7,000 Daltons. So it's actually a combo purification method. So as I mentioned, is the base resin is this 7,000 Zeba size exclusion resin, and so that 7,000 is your exclusion limit. So anything smaller than 7,000 will go around the resin, go through the resin and will be retained at that top band. Whereas anything larger than that will go through. So it's all about what's the size of the molecule you're trying to purify. So if you were trying to purify, like a smaller protein, 7,000 is that general rule of thumb. But the beauty of the Zeba, the biotin and dye binding columns, those have ligands that are specifically designed to capture small molecule reagents. So you really just, as long as your biomolecule is larger than 7,000, that's the main thing. But we do actually have in our PIS, the product insert sheet, and we do detail out the different reaction conditions the things you want to keep in mind when you're using different biomolecules. So that 7,000 is a cutoff for the resin itself, but due to the additional ligands, we recommend that a general rule of thumb is a 50,000 Dalton biomolecule is kind of a good thing to keep in mind. We find sometimes, depending on your modification of that biomolecule, sometimes the ligands will hold on a little bit more to those modifications, but it's all stepped out in the instructions. But generally speaking, the sweet spot is anything over 50,000 works exceptionally well, and you get excellent recovery. Going below 50,000, it still works well, but there's a little bit more considerations given what is your fluorophore you're using or what is the small molecule that sometimes can alter the retention times. But in general, that's a good rule of thumb is 50,000 or higher.
Okay, thank you. Our next question is, do you provide any fluorescent labels in the SWIR spectrum?
So the farthest we go out is the near IR. So we have, for small molecules and polymer dyes, most of them will go out to around 800 nanometers. That's not really SWIR, that's really more near IR. That's a good question though. Actually, I can follow up by email on that to give a more thorough answer.
Perfect, thank you. Next question. I have a problem washing a DBCO dye from fixed cells. Any suggestion for wash steps?
So the DBCO reaction itself is extremely specific. It's bioorthogonal, meaning it won't cross-react with anything in the cells. So what that's meaning, if you're having a DBCO dye that's sticking to your fixed cells, sometimes it's not coming from the handle itself, it's probably coming more from the fluorophore. So sometimes if you have more hydrophobic dyes that are just stickier dyes in general, that could be the problem. If you have the azide modified, right, so the other partner in that reaction, if that is having non-specific binding, then the DBCO dye will follow it. So you really want to keep an eye towards where is your azide label? Is there potential cross-reactivity on the azide steps, because the DBCO dyes will just react wherever there's an azide. So if you have off-target azide modification in your cells, then you'll see that either DBCO reacting to those off-target azides and coming up as background, or it could be that the dye itself is just non-covalently sticking to your cells and isn't washing off. So you could look at the fluorophore itself and see if you've got a stickier fluorophore. In general, most of the electrophores are pretty not sticky, so it's not usually a problem. But yeah, I would look at that, look at the fluorophore, and then also look at where is your azide modification happening, and is that specific enough for you?
Perfect. It looks like we have one final question. Have you done antibody with oligonucleotide? Yes,
Yep.
oligonucleotide. What is the bind site for it, and how to evaluate the conjugation result?
Yeah, so that's a good question. So oligonucleotides actually can be conjugated with a lot of different methods. It just kind of depends on what it follows the same thought process for a general conjugation. You're just thinking it comes with a little bit more background that you have to consider. So like, what's the size of the oligonucleotide? How do you want to attach the oligonucleotide? So do you want to use amine chemistry, cysteine chemistry? There's lots of different ways you could do it. It just really depends on what type of modification do you want to put on your oligonucleotide. So like, for example, you can have an amine group on your 5' end, for example, of your nucleotide. You can use that for amine targeted cross-linking. You could have a sulfhydryl group modified. There's lots of different ways to do it. In general, you treat the oligonucleotide as the label, and you follow it. It's compatible with a lot of the same chemistries. The only thing you have to consider is it's a little bit of a different thought process as far as how might that affect your protein. So you're putting a large negatively charged string onto that antibody, so you have to design. You want to try to keep that degree of modification relatively low. And then you also have to be wary of, you're purifying, instead of purifying out small molecules, you're purifying out larger oligonucleotides. So depending on the size of that oligonucleotide, your purification strategy might have to change. Generally speaking, in most conjugations, your protein or your biomolecule of interest is significantly larger than your modification. So size exclusion chromatography works really well to separate those out easily. If you're starting to think about larger modifications, like whether protein or oligonucleotide, you have to change your purification strategy because your modification might be similarly sized. So oligonucleotides can range obviously based off of their sequence length. So if you have a really short oligo, then that might be pretty easy to react and pretty easy to purify. But if you have a longer oligonucleotide, that might be as heavy as your protein.
All right, thank you so much. Doug, do you have any final comments for our audience?
I actually see one last question popped up, and I'm happy to answer if we have some time.
Sure, yeah. Let me pull that up. Okay, so final question. Can the Zeba columns be used with Q dots to remove linking molecules?
Short answer, yes. They work great, actually. So the Q dot modifications, Q dots most workflows are using the heterobifunctional crosslinkers. So the Zeba columns work fantastic to remove those linking molecules, and the extra weight from the Q dots makes it elute really nicely from the Zeba columns.
All right, thank you so much. Do you have any final comments for our audience?
The final comments would just really be, we're really excited to bring a lot of new technologies to making your own bioconjugate and how to design and buy a conjugate or shop for one. It's never been this easy to make your own conjugate. So if it's something that you're interested in making your own tools for your experiments, it's never been easier to make your own reagents these days. I wish I had all these tools when I was in grad school. It certainly would have made my life easier. And I think what we have on offer will certainly make your life easier too. So hopefully this was useful, and I'm happy to answer any questions by email for follow-up. Thanks, everybody.
Thank you again, Doug, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.